Welcome once again. In our previous video on mutations, we had seen the various types of mutations that are possible, the various inheritable changes, sudden changes in a DNA sequence, in the sequence of polynucleotide stretches in the biological world. Now, let us try to understand these inheritable uh, changes, sudden changes on a molecular basis, that is at the level of muton, the nucleotide in this present video. So these mutations, they can result either from the replication errors, that is at the time when the DNA is being replicated, at that point of time certain changes may take place in the nucleotides, that is the unit of mutation, or they may occur when certain the gene elements, they transpose themselves, that is because of the presence of jumping genes, transposable elements. In nature, certain spontaneous lesions may be generated, which leads to these inheritable sudden changes. So they are the intrinsic factors present within the cell system that leads to mutation. And at times, there are certain external factors, the extrinsic factors, which may be physical factors or they may be chemical in nature, they may be different chemicals. These extrinsic physical and chemical agents that result in these inheritable sudden changes in the DNA sequence, they are known as the mutagens. So these mutagens, they and also the spontaneous mutations, these mutations, they may be studied at two levels, or they may occur at, they may be studied at two levels. First is at the DNA level. Now here we are using the word DNA because this sequence, the change, the alteration, it may be within the coding sequences or genes which are expressed at the protein level, or these mutations, if they take place in the regulatory gene sequences, they may affect the process of transcription itself, that is formation of RNA from that particular stretch of DNA, or at times these mutations, they hinder the translation process, because if the mutation is taking at some point which will help in the uh, joining of this messenger RNA formed with the ribosomal machinery, or at some other point of translation, that is for the formation of the translation complex, if they are contributing and they get mutated, then they affect the translation part of the central dogma. And there are certain mutations which take place in the non-coding sequences, means neither they affect the proper coding sequences nor the regulatory sequences. Now this type of mm, uh, mutations in the non-coding sequences, they are not expressed, and mostly either they amend themselves or they just accumulate within the genome, in the gene pool. And these mutations, which are basically taking place at the level of nucleotides, at the level of DNA, they are expressed at protein level. That expression, it may be either in the form of alteration in the basic amino acid sequence of the protein formed, that is the genes, or at the transcription level. At the transcription level, the rate with which the particular protein is formed, that is altered. And that is the level of expression. Or at the translational level, that whether the messenger RNA formed is capable of being translated or it is unable to associate itself with the translation machinery. So these mutations, whether in genes or in regulatory sequences, they are expressed at the protein level. Now, how these mutations take place? What all happens within the nucleotide sequence that leads to the mutations? So, these mutations, they can basically either replace a base in the DNA molecule, or they may alter a base so that it mispairs with another base, and at times there's a damage in the base so that it can no longer base pair with any of the base under normal conditions. So that leads to mutation of that particular nucleotide sequence. Now, at DNA level, we say that nucleotide is the basic unit of mutation. So, if this substitution of this single nucleotide, now we say single nucleotide, but actually, eventually, it is the base pair that is substituted. Because just now we'll see how. So, at DNA level, if this mutation, it can be of two categories, as we have already seen in the video on the types of mutation. They may either be the transition, in which the purine is replaced by another purine, or a pyrimidine is replaced by another pyrimidine, or it may be of transversion type, in which a purine is replaced by a pyrimidine and vice versa. So, whether it is a case of transition or transversion, eventually the base pair, it gets substituted. That is, the AT pair, it is replaced by the guanine cytosine, because if adenine, it is substituted by a guanine because of any reason, in that case, in the second replication cycle, this guanine will pair with cytosine. So now the AT pair, it is now replaced by the GC pair. So the transitions and transversions, they lead to base substitution. That is the substitution of the adenine thymine pair with the cytosine guanine pair. Now at protein level, what is the impact of this nucleotide substitution or change in the nucleotide sequence at protein level? Now, if we consider the transition transversion case, so this single nucleotide base pair substitutions, they lead to either the silent mutation or the missense mutation or the nonsense mutation. This we have already dealt with in the types of mutations. But here we are trying to co-link what happens at, as a result of the particular mutation or change on the nucleotide level. So if the change is of the sort that it codes for the same amino acid, because we know that genetic code is degenerate. So arginine is coded by both AGG as well as CGG. So if here A is replaced by C, there's no change in the protein that results, because even the mutated triplet, it codes for the same amino acid. Such type of mutations, they are known as the silent mutations, or they may be missense mutation. That means because of the mutation, some other amino acid is being coded. If this other amino acid is chemically similar to the amino acid that is being replaced, like a basic lysine is being replaced by basic arginine, the central A is substituted by this guanine. So such type of missense mutations, they are the synonymous missense mutations. Whereas if some dissimilar amino acid is produced because of the mutation, then it is known as the non-synonymous missense mutation. And this type of mutations, they alter the basic nature of the protein itself. For example, if the hydrophobic phenylalanine, which is coded by UUU, it is replaced. Here the central uracil is replaced by cytosine. So it leads to the formation of a polar amino acid, that is serine. Now the next type that we had done was the nonsense mutation, in which because of the substitution of a particular nucleotide, like the cytosine in this case, it leads to the formation of the termination codon. That means the functional gene, it terminates before acquiring its complete amino acid sequence, and that leads to nonsense mutation. Now instead of being substituted, that is instead of undergoing the transition or the transversion type of mutation, if there is addition or deletion of a single nucleotide base, in that case frame shift mutation takes place. And by frame shift mutation, we know that the triplet in which the amino acid, it reads the codon present in the messenger RNA, it alters for all the consecutive amino acids that will be incorporated on that particular mutated nucleotide sequence or the messenger RNA produced on that DNA molecule or stretch of nucleotides. So this type of addition or deletions, they always, wherever there's a frame shift mutation, it always leads to a, alteration in the amino acid, which may be subvital or lethal and to the organism. So how this occurs? Now we know that all these nitrogen bases, that is the adenine, thymine, guanine and cytosine, they basically may occur in the nature in both the tautomeric forms, in the keto form as well as in the enol forms. But in DNA molecule, these nitrogen bases, they are present in the keto form, And in the keto form, cytosine, it forms triple hydrogen bond with guanine, and thymine, it forms two hydrogen bonds with adenine, and these combinations, they form the stable base pairs. But if there is a tautomeric shift, that means the keto form, it converts to its enol form because of the change in the electron pairing of these particular nitrogen bases. So what will happen is the complementary base pairing of these enol form of nitrogen bases, that will alter. For example, if cytosine, it undergoes tautomeric shift, instead of pairing with guanine, it will start pairing with adenine. Similarly, the enol form of thymine will pair with guanine, or the enol form of adenine, instead of pairing with thymine, it will start pairing with cytosine. And the rare enol form of this guanine, instead of forming the three bonds with mm, cytosine, it will pair with thymine. So, in this manner, because of tautomeric shift, the complementary base pairing of the DNA molecule is disturbed. In the first round of replication, suppose say here in this case, if this enol form of guanine, it will pair with thymine. So when this GT, it will undergo replication, the DNA having this combination of base pairs. When it will undergo replication in one of the strands, it will be guanine pairing with the regular cytosine. But in the other strand, this thymine, it will pair with adenine. So the initial guanine cytosine base pair is being replaced by the adenine thymine base pair. In the parental DNA, if DNA replication takes place, these A and T, this thymine, which is base pairing with this enol form of guanine, and when this stretch of nucleotide, it undergoes replication, it leads to the formation of a mutant along with the wild type. So the gene pool will now have this mutant form of uh, stretch of nucleotide sequence also. Now another type of mispairing, it can also occur because of the presence of certain base analogues. Now base analogues, they are the molecules which resemble the nitrogen bases in their pairing with each other. For example, the bromouracil, the 5-bromouracil, it is an analog of thymine. That means this 5-bromouracil, it will pair with adenine in a similar manner like thymine, it pairs with adenine. Okay. But this particular base analog, it may be ionized. It has got these bromine, and because of this bromine, this oxygen which forms hydrogen bond, it will get ionized. So because of the ionized form of this base analog, the complementary base pairing, it alters. That is the 5-bromouracil, which pairs with adenine like thymine, that is the normal keto state. But if it gets ionized, it will pair with guanine. That means in the next generation, the AT pair, it will be gradually replaced by the GC pair. Similarly, there's another base analog, that is the 2-aminopurine, and this 2-aminopurine, it is an analog of adenine. So it pairs with thymine, but in its protonated form. That is when it acquires a positive charge over here. In that case, instead of pairing with thymine, it will pair with a cytosine. And now this AT pair, it will be gradually substituted or changed over by the GC pair. So this mispairing, it may occur because of these ionized forms of base analogues. Another point may be that the existing base, the nitrogen base, it is not replaced by any of its analog. Instead, there is a change in the basic chemical structure of the nitrogen base itself. So there are certain mutagens, chemical mutagens, like the alkylating agents, which alkylate the EMS or the ethyl methane sulfonate. It adds the ethyl group, and there's another alkylating agent, that is the nitrosoguanidine, which methylates, adds methyl. And it has been observed that this oxygen present on carbon number 6 of the nitrogen base, or this oxygen which is present at the carbon number 4 of the pyrimidines, they are more susceptible to alkylation. So guanine, it is converted to this ethylated guanine, and this ethylated guanine, it instead of pairing with cytosine, it will pair with thiamine. Similarly, the ethylated thiamine, instead of pairing with the adenine, it will pair with guanine. So in this manner, changing the basic chemical structure of the particular nucleotide, of the particular nitrogen base of a nucleotide, it leads to alteration of the base. That is, it leads to mutation. Another possibility is that there are certain mutagens like proflavin or acridine orange or ICR 191. These are very voluminous molecules, and if by any means they slip in between this nitrogen bases, they will, but at the time of replication, they will lead to deletions of the single nucleotide pairs. So presence of intercalated molecules within the DNA, that is in between the stacks of nitrogen base pairs, that leads to mutations. Now another possibility is that neither the base analog is involved, nor there is a change in the chemical structure of the nitrogenous base. Instead, there is a damage to the nitrogen base itself. For example, the ultraviolet lights, they induce dimerization of pyrimidines, both cytosine as well as thiamine. So these dimerization, it leads to lesions. So it interferes with the normal replication process of the DNA, and that leads to mutation. Or there are certain molecules like aflatoxin. This aflatoxin, which we know it is obtained from a fungi and is very toxic. So addition of this aflatoxin, it leads to the breakage of the bond between the base and the sugar. That is, it just separates the nitrogen base from the backbone, and this leads to the formation of a purinic site because this purine guanine is being separated from the backbone. This is the normal backbone with these nitrogen bases, and now if your aflatoxin is added to such a DNA, it will break this bond. So we will get a DNA molecule in which lacks one of the purine site. So this site is referred to as a purinic site, and this type of DNA molecule is known as depurinated DNA. So depurination at the guanine residues, it preferentially it induces the transversion reaction or transversion type of mutation. That means at the points from where guanine is replaced at the time of DNA replication, it that particular site, it will be taken over by this 80 pair, 80 base pair. So this leads to mutation. So we have seen that the various mutagens, they induce mutations by a variety of mechanisms. Some of them, they mimic the normal bases and are incorporated into the DNA where they mispair, while others, they damage the base pairs that are there, and either they cause a specific base pairing or they destroy the pairing by causing non-recognition of bases. The spontaneous mutations that occur because of the replication error or because of the spontaneous lesions, they are basically in nature, they tend to repair themselves. So it is a case where the nature, it tries to stick to its original sequence, and the method by which these damages, they may be repaired, there are many a variety of mechanisms that we will be dealing in the next video on DNA repair mechanisms. Thank you.